All right, everyone. Welcome to the Southern eDNA Society's first webinar of 2025. Today, Josh Koh will be talking about explainable multimodal machine learning using combined environmental DNA and biogeographic features for ecosystem biomonitoring. Let's first start off with a karakia, as I'm in New Zealand. This is just a traditional opening in New Zealand to wish for a successful meeting. Whakataka te ho ki te uru, whakataka te ho ki te tonga, kia mā kina kina ki uta, kia mā taratara ki tai, e hi ake ana te atakura, he tio, he huka, he ho hu, ti he moriora. So I just have a quick few announcements before we get started. Quick reminder, if you haven't renewed your membership, go ahead and do that soon. I think that people potentially be starting to be kicked off the mailing list in sometime in September. So be sure that you renew if you haven't already renewed. Additionally, there's a news call for the newsletter. So if you've got any exciting news, say you've won an award or got an awesome paper published, go ahead and email Martin DeBrouwer. We'd really love to highlight your work. We also have a webinar sponsorship open. So if you or your team or your company would be interested in getting your logo out there, reaching a big audience, we have some spots for webinar sponsorship. And our next webinar is slated to be on Friday, the 21st of November at 2:30 NZST or 12:30 AEST. This will feature Lara Dennis Roy and Martin DeBrouwer. Additionally, we will be recording this webinar, so turn your cameras off if you don't want to be recorded. And then the webinar will live on in our SeDNAs Knowledge Hub once we get that up, and it will likely be members only. Now, without further ado, I'm pleased to welcome Josh Koh. He is the Chief Technology Officer and Team Leader for the Drone Operations and Technology Solutions Team for the Office of the Supervising Scientist in Darwin. So Josh, take it away.
Thank you so much, Claire, and thank you for having me this morning here. I'm just going to try to share my screen. Let me have a look. Is it clear? Can you see the screen?
Yeah, looks all good.
Fantastic. I'd like to start by acknowledging the traditional owners of country throughout Australia. Specifically in Darwin, the traditional custodians are the Larrakia people, and based up in Kakadu, where the Ranger mine is located, the traditional owners are the Mira people. We should pay respects to their elders past and present. The webinar or talk today, I've structured it in 5 sections. To start with, I just have a few slides just to introduce the Office of the Supervising Scientist. And then we move into the actual talk. It follows pretty much a standard, format like a journal article: introductions, methods, results, and conclusion. So to begin with, the Office of the Supervising Scientist is a special branch within the Commonwealth of Australia that's entrusted with the responsibility of protecting people and environment of the Alligator Rivers region, including Kakadu National Park, which is a World Heritage Area, from effects of uranium mining. And OSS conducts supervision of mining activities, research and monitoring of environment, and provide advice to the ministers and general public regarding the safety of uranium activities up north here. And at the moment, the largest project that we have is the monitoring and evaluation of Ranger uranium mine rehabilitation, which was until recently one of the largest uranium mines in the world. It is owned by Rio Tinto, and the rehab is actively undertaken by Rio Tinto. It is expected to take until at least 2034 and cost more than 2.2 billion Aussie dollars. And as mentioned previously, OSS undertakes environmental monitoring and the drone operations and technology solution team, the DOTS team that I lead, provides support and leads in majority of the remote sensing platforms deployed. For environmental monitoring, we combine innovations from 3 disciplines. That's remote sensing, artificial intelligence, or AI, and omics, that's referring to DNA-based biomonitoring approaches. The 2 routine remote sensing platforms that we deploy are satellite-based platforms and drone platforms. And we also use proximal sensors, which are sensors deployed close to targets, such as underwater videography for annual fish biomonitoring, and collection of environmental DNA samples. Those are performed by the water and sediment quality team, which is led by Dr. Andrew Harford, which I believe some of you are familiar with. And just to give an idea, the kind of scale of monitoring we conduct, we hope to achieve monitoring of Ranger uranium mine at scale. And this is just an example of a satellite capture April this year across 800 hectares of land. And within specific plots, we have high resolution drone imagery, including collection of 3-dimensional data from a laser scanning sensor called LiDAR. And from that, we derive tree isolation height profiling. And from revegetated areas of the mine, for example, area called Pit 1, we also have computer vision models that segment and isolate individual trees, and we track them across time and space. So we recently just start to deploy our expertise in AI and software development to support the annual eDNA biometry program. Specifically, at the moment, we are working with Harvard's team to develop a bioinformatics software for processing of eDNA amplicons. We call this software Chimera, and I'm hopeful that we can release the source code and manuscript in a very near future. Which brings me to the, I guess, the topic or context of the talk today. That's the introduction to the webinar today. Before that, a quick disclaimer. This quote is by Sir Arthur Tansley, one of the pioneers of ecology. He's an English botanist. He says, to the lover of the prescribed routine methods with the certainty of safe results, the study of ecology is not to be recommended. And that sort of like tells you that all the methods and results that I'm going to present today is clearly unsafe. And to confess, I was telling Claire that I'm actually not a domain expert in the eDNA research domain, but I'm approaching the subject matter more from a machine learning perspective. So depending on the outcome of the talk, this could be my first and last talk on this subject area. Just as mentioned previously, we're interested in the environmental DNA, eDNA, biomonitoring. And this is just a pretty picture I've just lifted off from the CSIRO website showing the wonderful applications of eDNA. Now, eDNA has been described as a complex mixture of genomic DNA from many different organisms found in environmental samples. But I think it has since been extended to include various broad spectrum physical forms that can exist, whether in filaments, tissue, fragments, whole cells, or whole organisms that you retrieve from environmental sample that will include soil, water, air, etc. I guess the premise of eDNA is that you would amplify an environmental sample using specific primers targeting known genetic markers, and you would retrieve sequences and you would match it to a known sequence of a species in a, for example, a database. The idea is that you could retrieve multiple taxas concurrently in a high throughput and cost-effective manner. So it's used now increasingly for, I guess, environmental assessment and biodiversity assessment as well. But there's a huge but here. A couple of challenges with the eDNA metabarcoding approach. The first one is dependent on availability of reference sequences for taxonomic identification. It suffers from biases in amplification, both in the initial PCR and also sequencing biases. And studies have shown that there could be various degrees of agreement between taxonomy retrieved using the eDNA approach versus traditional morphology-based approaches. But by far most, I believe, the greatest barrier, for the application method is the lack of reference sequences. And unfortunately, this is a challenge or problem that AI or ChatGPT can't solve. I don't see it solving it anytime soon. And so this led to some experts has come up with a different approach of utilizing eDNA data. They propose the de novo approach, which essentially is an approach advocating for methods that utilize intrinsic or inherent information capture or present within the molecular structure of the eDNA. And one of the approaches proposed is a machine learning based approach. And this is a taxonomy-independent approach. And I need to make the point that this approach is not to, supplant or compete against a taxonomy-based approach, but rather I see it as a complementary approach to the existing taxonomy-based usage of eDNA. This is just one of the papers that I've come across when I was researching about the status of machine learning in eDNA. One of the earliest studies by Tristan Cordier is that they used eDNA to predict marine biotic indices. These are indices that's calculated as a proxy to indicate certain quality gradients, for example, water quality, based on eDNA meta-barcoding data. And they show that it's comparable to morphology-based approaches. And subsequent studies have used machine learning to predict biotic indices in freshwater systems. And also they show that the values inferred by machine learning sometimes outperform taxonomy-based strategy. And this sounds really promising. And somebody was considering ML. They come up with some considerations or challenges in the ML applications. Some experts noted that there are certain limitations with how ML is being deployed at the moment. Specifically, the use of ML to infer traditional indices also inherits the error and biases of the old system. Specifically, what the concern is when you use ML to, predict a singular indices or couple indices, you are actually not harnessing the full potential of the high-dimensional, high-throughput eDNA data for environmental assessment. So the question asked was, can we use ML to directly infer environmental state of quality gradients? Unfortunately, in recent years, a recent study by Freshwater, they used a combination of machine learning and eDNA to directly infer reference impacted state of 64 federal freshwater biomonitoring sites in Switzerland. The model used was random forest, coincidentally, probably the most popular machine learning model used at the moment in the eDNA studies. What was used was a OTU feature table. Now, OTU, most of you familiar within the domain would know that that's the operational taxonomic unit. Essentially, these are sequences or consensus sequences that are representative of clustered sequences, a group of sequences based on sequence similarity, most commonly set at around 97% similarity. So they use the OTU feature table as input extracted from macroinvertebrate eDNA as input into the machine learning process. And they found that the random forest model performed better with about 69% accuracy compared to a model trained on traditional taxonomy data. And they made the conclusion that eDNA alone is enough to be used in an ML process to directly infer environmental states. However, I make the observation that the results reported, the 69% accuracy, is based solely on a training data set. I also make the observation across some studies that machine learning performance seems to be reported purely on training data sets. And the usual practice is that you would train a machine learning model on a training set and evaluate the performance using unseen data or test set. So this implies that potentially the 69% accuracy could be lower, which is usually the case when you deploy trained models on unseen data. And if you ask me, 60+% is a good start, but I think it leaves room for improvement, which brings me really to some observations I have on the current state of the ML approach in the eDNA research, which is the first challenge is ML could lead to quite a huge variable in performance. In classification tasks, which you assign labels or category to the samples, you could have anywhere from 50 to 80 plus accuracy, a huge difference. And I think some of the limitations could be due to the use of models, specific ML models. This is purely my own personal observations that most studies mainly use classical models. I refer those to as support vector machine, artificial neural networks, or random forests. And as opposed to more current, I guess, state-of-the-art gradient boosted machines such as extreme gradient boosting machines, XGBoost. Partially, I think it's because it requires technical expertise for optimization. So technical barriers to adoption of different, I guess, more advanced models could be one of the reasons. But also the input used in most of the current machine learning studies involving eDNA is that the eDNA is most certainly the only input. And that has some limitations because environmental processes are pretty complex. And depending on your target, which is the next point, eDNA alone might not fully account for all the drivers, complex drivers, responsible for the ecological process of interest. And so, this is just some of the observations that I think would impact on the performance of the machine learning approach. But this is not the most challenging, because it is quite possible for domain experts to optimize specific custom models to increase performance. I think the greatest barrier or challenge in the ML approach in the eDNA domain is the number 2 point, which is there are concerns of reproducibility and transferability. For example, it is my observation that the majority of studies uses what I call variable feature, and that's really, I guess, that's referencing the OTU or the operational taxonomy units. Those are essentially, in my opinion, variable features, because they could change depending on your data sets, and different data samples would potentially result in quite different sets of OTU groups or clusters. And that would, I guess, make reproducibility quite challenging. And coupled to that as well, I mean, most of the machine learning procedures are either not, I guess, shared, not easily shared, although there are descriptions within most papers of the methods, but the codes are either partially available, or they are made available without the full training data set, again, making the workflows hard to reproduce. And finally, second point is explainability. That's, how do we understand what the model is doing and which features important for the model in the machine learning process? And this is crucial to ensure, I guess, trust and confidence in the ML approach. At the moment, it's either not well explored or limited to feature importance. I think this is also linked to the fact that the most popular model at the moment is the random forest model, in which it's quite easy to compute feature importance. But caution needs to be exercised when we are using feature importance, in that it will pick up features important for the model, but not necessarily having any biological or ecological significance. And so, with that in mind, I was having it thoughts around designing, potentially exploring different methods based on my knowledge in machine learning approaches to potentially address some of the shortcomings. And obviously, this is can be purely subjective, but this is where the talk goes into. That's the workflow that I've been, I guess, slowly chipping it away. And I call it explainable multimodal ML workflow. And it's based on two, I guess, trying to address two challenges or guiding principles. The first one is I wanted a workflow that could provide good out-of-box performance. And this is very subjective, but for example, I'm hoping that it will give me close to 70% classification accuracy without much effort as a general principle. And a workflow that allows rapid prototyping of state-of-the-art ML models. So we're talking about a modular pipeline where we can have components we could plug and play or replace as more advanced algorithms becomes available. And I'm hoping that this workflow could be compatible with multiple data modalities. As mentioned previously, eDNA being the sole input has limitations. And with the explosion of next generation sequencing and high throughput technologies and other data modalities such as remote sensing data coming in, I think we are well posed to harness multiple data types in our workflow. So I wanted a workflow that could be compatible with multiple data modalities. And it needs to be easily adaptable for, I guess, classification or regression tasks. And the second point, which the most important point as the guiding principle of the design of the entire workflow is it needs to be reproducible, transferable, and explainable. And in, I guess, some design choices was made to emphasize this point. For example, earlier on, I've made the choice to adopt exact sequences as the base unit of analysis. That means adopting exact sequence variants or ASVs as absolute references. And second of all is to consciously only adopt any data manipulations that can be transferred across data sets. For example, any manipulations like normalizations or transformations need to be transferred across data set. So to ensure that it is independent of the data set. And this next point is it has to be in a pipeline that can be shared easily in standard formats. And finally, we will adopt explainable AI techniques. These are common techniques in machine learning that's deployed to explain model behavior, not only which feature, but how are the features impacting on all the predictions. And with that, this is what I've come up after I've, moments of confusion and clarity. This is sort of the step that I've cobbled together. And so there's 5 steps in this workflow. The first step is a feature selection and dimension reduction step. Now, none of the steps are really safe here. These are unsafe methods. The reason that I start with feature selection, dimension reduction is that ASVs data sets, big exact sequences are typically quite large. It can range from tens of thousands to hundreds of thousands or million sequences in very large projects. And to make them, I guess, that high dimensionality makes them not really amenable for merging with other data modalities. So my first step was to reduce the data size and select high quality features. Now, I should mention that other common feature selection techniques in machine learning, such as feature importance or statistical-based approaches such as univariate filtering, would also work to reduce and select features. The reason I go with a different approach is that I wanted a feature selection that will be biased towards selecting features or ASV that could have potential biological significance or ecological significance. And therefore, I've chose to adopt what a technique called differential abundant analysis, which are essentially a series of statistical approaches or machine learning approaches that identify ASVs or features as differentially abundant across the environmental states of interest. And so I start with this first step. And the second step is really just further filtering of the differentially abundant ASVs using log-fold change. A log-fold change essentially is a parameter to indicate the magnitude of change or differential abundance of a feature. It's a proxy potentially to the magnitude of signal that is the feature could contribute. And so by filtering out the lower features that have lower fold change, we are essentially retaining the features which potentially has the highest signal. And the third step is I would merge then the resulting selected filtered ASV feature table with additional data coming from what I call biogeographic data. But essentially, this could be any data in a tabular format in columns, could be a text data, could be a numeric data. And what I did was just append the columns without further manipulation onto the ASV table, feature table. Essentially, this is possible because the step number 4 is really where everything comes together, is that I employ automatic machine learning systems to do all the heavy lifting, to pre-process the features, to optimize the models, machine learning models, and to do the selection. And finally, the resulting machine learning models are explained using a feature-based explanation method, which is based on Shapley values. These are values to show how much a feature is contributing towards the machine learning prediction. And it's a very popular approach now, common in machine learning practices. One of the packages available is called the shape values. Can be used to explain the models, not only which features important, but how are they impacting on model behavior. So as a whole, this are the general idea of the steps and the data sets. So once I have some sort of workflow, the next thing is I want to test the idea. Now, being a non-domain expert, I do not have existing data sets. Fortunate for me, in a previous paper, Francois Keck's paper, when I went to his GitHub repository, I noticed that not only did it have information for the 69 sites, it actually have information across the entire Switzerland for the eDNA sampling data. So thank you, Francois. And in it, I've managed to retrieve 63 sites of biomonitoring sites that have eDNA samples. In total, from his paper, I managed to retrieve 23,600-ish ASVs. And with it as well, Keck provided taxonomic annotations of all the ASV based on their internal curated database of macroinvertebrates sequences. And the paper as well contained a detailed site metadata, including location, altitude, the biogeographic regions, etc., for across these sites. And so I've followed the criteria to classify all these different sites based on the notion that any land proportion more than 33% of natural land, i.e. forests, are classified as reference, and those having less than 33% of forest area are considered impacted due to urbanization or agricultural activities. And these are just a distribution of the site across Switzerland, across the 6 major biogeographic regions. And that's the data sets that is used in my study. So the plan is pretty straightforward. It's to design first to split it into a training and test data set. I use a 80 to 20% ratio split. From the 163 sites, we have almost equal distribution of reference impacted sites. And however, I've noticed as well within the data sets, there's 2 sampling seasons across summer and spring. And most of Keck's study was occurred in the spring sampling. Each of the sites have 4 replicates, but some of the sites have half of them coming from, for example, summer, and 2 from the spring season. The safe approach will be just to stick to 1 season, but since we're doing unsafe stuff here, so I've decided to take everything in. And to account for the seasonal changes, I've encoded each of the site as a site season. So for example, you can have site A underscore summer as an individual sample and site A underscore spring as another independent sample. And with that, I've ended up with 223 site seeding samples. And if we do an 80-20 split, that's 189 training and 48 tests, unseen test sets. And it's important to mention that all the subsequent results that I'm going to show are based on the unseen data for the test samples. So once we have a training and test data sets, the next step I did was to establish a baseline machine learning performance using eDNA data only. The task we're going to do is to classify each site, predict whether it's a reference impacted. Now, this seems a fairly simple classification task, but given the complexity of, I guess, heterogeneity of the data across different biogeographic regions, across different seasons, this could be, quite a challenging task. So I've derived from the eDNA, starting with the ASV data sets. I've clustered them using a research algorithm, which is a very popular standard practice now, using standard default parameters into OTUs. And then from the ASVs, I perform differential analysis using two, again, very popular algorithm in the research domain, which is the encompassing and differential ranking. So I've ended up with 4 data sets, which is the first, the raw ASVs or unfiltered ASVs, the OTUs, which is a clustered form of the ASV, and 2 subsequent ASV, which is selected using the differential ranking, differential abundant analysis. And the machine learning, automated machine learning package that I use is PyCaret. I need to mention as well that all this experiment machine learning framework was implemented in Python. And PyCaret is one of the, again, fairly well-known AutoML package, which allows the user to define individual components. And so the 6 machine learning algorithms that are defined to automate is the support vector machine, the random forest, and then we have the trio of gradient boosted machines. These are the more recent ones, the light gradient boosted machine, the XGBoost and category boosting, CatBoost, and followed by multi-layer perceptron, which essentially is a form of artificial neural network. And once we have a baseline performance, I explored potential of further feature filtering by, I guess, as mentioned earlier on, by filtering out features that have less differential in magnitude. And so once we have some sort of baseline, we can then begin to evaluate what if we add further information from, for example, biogeographic features into with the eDNA data. The biogeographic features that was added was the biogeographic region, that referring to the previous 6 major domains across Switzerland, the catchment area and altitude of each of the sites. Now, the reason these 3 features selected is because they are the only first look, they are the sensible features, and also they are the only features that is, I guess, complete across all the 163 sites. And the multimodal data set is combined by selecting a filtered ASV from previous steps with 3 of the columns, the biogeographic region, catchment area, altitude, just appended onto it as a feature table. And then I would repeat the entire ML experiment for the 6th machine learning model using the multimodal data set. And finally, it's pretty straightforward, just to explain, calculate the shape values for machine learning model on the training data set. Now, the normal practice is to calculate shape values on training data set. You could also do that for a test set, or some would combine tests and training. But it doesn't really matter, because the whole idea is the shape values as a proxy to indicate model behavior. And the usual practice I need to mention is we usually calculate the shape values on a training data set. And to show the top 10 features detected based on the shape values, there's 2 forms of popular visualization. One is the bar chart, one is the B-swarm plot. I will explain further as we go through the results. And finally, to incorporate taxonomic information where available for some of the ASV feature, for example. And now we move on to the results So the first thing we did, or I did, was to establish a baseline. That is to do impact prediction using eDNA only. And as shown in the bar chart here, we have the 6 machine learning models. And underneath the x-axis is the data type. So the first one is the unfiltered ASV, or the raw ASVs. We have 23,000 of them. And second one, after clustering, we have the OTUs. We have about 6,400 OTUs. But interestingly, or shockingly, this is where things get really unsafe, is after ANCOM BC selection, ANCOM BC picked up only 293 ASV as being differentially abundant across reference impacted site. The second approach, the differential ranking approach, identified far more ASV as differentially abundant, about 1,339. And if you look across the bar chart, we can see that starting from the ASV, we see that performance is about 50 to 66% accuracy. For the OTUs, it's about 50 to 64%. And for interestingly, for the ANCOM BC, which only had 293 features, it still put in a very respective number of 54 to 64%. And lastly, differential ranking picked up the best performance, resulting in up to 68.8% accuracy. And that's achieved using the multiple layer perceptron model, MLP model. And across the board, we can see a general trend in terms of the ML model performance that support vector machine is the most, I guess, perform the poorest. And then subsequently, performance of increase across as we move across from random forest to the more current gradient boosted machines, and finally the artificial neural network MLP. What is interesting to me is that between the ANCOM BC2 and DR data sets, so differential ranking contain all the 293 features picked up by ANCOM BC2. Suggesting that ANCOM BC2 is a much more stringent selection process, at least on this data set. But it also suggested to me that there is still room for improvement in terms that we could filter further. So we have some room to wiggle around between 293 features and 1,339 features. We could deploy our feature fusion strategy, which essentially the next step I did was I focused on the ASV from the differential ranking, and I perform a couple of filtering, but also I include 2 of the best models, which is the MLP and the category boost model. And that's the feature filtering that I performed. So what I did was essentially did a percentile filtering, starting with 0 percentile essentially is unfiltered. 30th percentile is actually removing anything using 30th percentile as a lowest threshold, and then 40th and 50th percentile. And just by doing this simple practice, we can see the impact on the 2 best performing models in that both MLP and CatBoost had the best performance at 40th percentile threshold. And so, and then 40th percentile is determined as the optimum threshold. I need to mention that the filtering needs to be, the optimum threshold needs to be determined empirically for different data sets. But at least for this practice, 40th percentile works really well. And at 40th percentile, not mentioned there, the number of ASVs is clocking in about 800 plus ASV, which is a pretty significant achievement considering that we started with 23,000 ASVs, now we're down to 800 plus high quality ASVs. And so just a quick glimpse at the workflow. The workflow is can be easily retrieved from the pipeline. One of the reasons that I've selected PyCaret is because it saves entire workflow, all the parameters in a scikit-learn format. Scikit-learn format, for those familiar within the Python data science, is one of the considered the de facto machine learning package, and the format is widely adopted in, I guess, the Python ecosystem, and that's why one of the reasons I chose PyCaret as the package of preference. And so from the pipeline, we can easily retrieve all the steps. And this is quite standard and common across all the 6 machine learning. PyCaret, step 1, it does something called label encoding, which assign the numbers 0 to impact the states and 1 to reference. And step 2 and 3 is not really relevant, as it's just an automatic process in which it tries to impute any missing data which we don't have. So it moves straight on to step 4, which is it will deploy the machine learning model and optimize it in the process. So the training happens across 100 optimization rounds, and it's trained using a tenfold cross-validation approach. Essentially, this is a very common practice again in machine learning, in which you would divide a training dataset into certain partitions. For example, tenfold indicates that you would divide it typically into 10 partitions. And in the first round, you would reserve 1 partition as a test data set, and the remaining 9 partitions will be trained as a training set. And for the second round, you will do the same thing, repeat. Different partition will be reserved as a test set, and the remaining 9 will be used as a training. And this repeated until 10 folds or 10 rounds finish in the training. So this is the entire workflow. And I want to show the model explanation using shape values for the best model that we retrieve in our, following our feature filtering, which is the MLP at 40th percentile log fold change thresholding. The first plot is the bar plot. Then this is the easiest plot. Everyone loves this because it's just like the feature importance that you get from random forest. It is based on the mean of absolute value of shape. Essentially, just show the average impact of its features on your prediction, overall model performance. And you can see the, for example, the Beatis rodani is the top performing feature with the highest relative contribution or impact on the model. And this is essentially very easy to have a look of which are the important model. The second plot, which is arguably perhaps the most important plot generated by SHAP analysis, is the B-SWAN plot. And shown here is a B-SWAN plot to show the impact of these features on the reference class. You could swap it around for impact class, in which the I guess the diagram would just flip over. But either way, shown here is a B-SWAN plot for reference class. Now, just to explain how do we interpret the B-SWAN plot. So B-SWAN plot is ranked by the features ranked by importance. So you can see that it reflects the same ranking order as per the bar plot. And secondly, a features value or ASV abundance, relative abundance in this sense, is depicted from blue, which is a low value, to high, which is a red dots. And if higher feature value, the higher the abundance of a feature results in higher positive shape value on the x-axis. The higher positive shape value indicates that it has a higher probability of being assigned the reference class. So this implies that higher abundance, which is linked to high positive shape value, is showing that the features having a positive impact on the class prediction or positive association with the class prediction. And based on this, we can see that there is 6 features that having a positive impact on the prediction of reference class, with 4 having a negative impact. And this ASVs has been matched with the taxonomic information. And I've also did a further, I guess, verification using a BLAST, just NC by BLAST search, and to ensure that all the sequences match up correctly. And since we have an identity of specific ASVs, and this is the beauty of working with exact sequences, we can start to do literature research or review on the potential ecological or biological impact on these features. And this provides another line of evidence or validation of the SHAP analysis. And shown here is the red dots indicate literature study or results that support this specific species or ASVs as having associated with impacted states of environment, for example, degradation of natural environment or urbanization. For example, the first species, Baetis rodeni, is one of the most widespread mayfly species in Europe, and it is typically associated with urban-centric regions. And many research has even proposed it as a bioindicator for water quality or even degradation or urbanization status. And the second species, Autocladius telechaetus, is typically found in cold water, fresh, pristine water region, and it's very sensitive to temperature. So again, associated with reference states. And so if we line them out, compare them to the known effects shown by the ship plot, it's encouraging to see that they are congruent or in agreement with what the shape analysis is telling us. So again, this also is another validation of the use of differential abundance analysis in the first step to select potentially biological significant species, and resulting in, I believe, in the current results here, showing again that DAA, or the differential analysis approach, do yield merit in our workflow. And so once we establish some baseline at this stage, so the best model we have is about, 70.8 accuracy. That's the MLP model. Now the question is, can we achieve higher performance when we combine or introduce more features? And shown here is the performance or classification performance comparing eDNA only, which is using the filtered data sets ASV resulting from the differential ranking selection, and the same data set combined with the three biogeographic feature as mentioned previously. Now, the first encouraging note is that across the board, the multimodal data set yielded performance enhancement across all 6 models, has a significant performance uplifts. And the best model is the MLP, achieving 83.3% accuracy. And this represented a very significant uplift, about 12.5%. And the tree-based model as well, putting a very strong performance, 72.5 to 75%. And if we look at the pipeline, the only thing that has introduced in addition to previous pipeline is step number 4, which is the one-hot encoding. Essentially is just a simple transformation of a categorical feature or column by encoding absence or presence. This is one of the ways to, I guess, to convert categorical data, text data, into a machine learning friendly data. The effect is shown at the table. For example, you have the ASV initially just combined with 3 of the features, which is altitude. Numeric. This is a numeric feature. We have a biogeographic region, and you have the catchment and status. And so with the categorical encoding, it just converts each of the feature within the column, for example, biogeographic region of Central Plateau as a column, and it will mark it as absent presence across the sample. And by this, we essentially converted a mixed data type table into a machine learning friendly format. And so if we look at the shared values or feature importance across all machine learning model using the combined multimodal data set, we can see that it is actually being consumed in the actual feature learning machine learning process. Specifically for the multi-layer perceptron, which is our best model at the moment, 6 of the top 10 features are actually biogeographic features. And as we move across the tree-based model, the CatBoost, XGBoost, like GBN and random forest, we see that the top 2 features are biogeographic features. The top 2 biogeographic features is the Central Plateau as well, and altitude. But when we move across support vector machine, we notice something very interesting, that the altitude is by far the highest feature. And this essentially just indicate that it struggles with feature scales. In features that have huge I guess magnitude differences. it will, I guess, be having an adverse impact on certain models. And support weather machine is certainly one of the models and very sensitive to feature scaling. And some of the, you might ask, why don't we just then normalize scale this feature altitude again? This is a conscious design choice at the beginning of the workflow that we would avoid, I guess, deploying any manipulations that is dataset dependent. So in some sense that this entire workflow would preference machine learning models that are invariant or insensitive to feature scales. And so finally, I will show just the shape values on the multi-layer perceptron, our best performing model. And the bar plots, again, feature importance, showing the top 10 features that's important. From these top 10 features, we have 6 biogeographic features, indicating that, at least in this data set, the MLP is really performing really well, consuming different data modalities. And with the remaining 4 top features coming from the ASV or eDNA data. And out of the 4 ASV, 3 of it previously as well picked up in the eDNA only approaches in the MLP, with 1 new ASV, which is the NOCA species, picked up. And the top 2 features are the central plateau and altitude. And based on previously whether the abundance relates to higher probability class predictions, we can see that there are 6 features we have having positive correlation with reference class and 4 negative. And we do a further literature research. It shows that it's, at least in the central plateau, is one of the smaller regions in Switzerland, but it has 1/3 of the population and is the highest area with urbanization and agricultural activity. So it makes perfect sense that the number 1 feature is being picked up is by Central Plateau. And the other biogeographic regions, the Jura Mountains and Northern Alps, are quite associated with reference sites because they are less, they are more pristine rivers and less impacted sites on these regions. But importantly, the new ASV picked up, the NOCA species, is a known bioindicator for really high quality, pristine river systems. So again, when we line up the actual literature support for these features, it again is in good agreement with the Sharp value plots. So in conclusion, I believe the DA in combination with Sharp analysis is a powerful combination of tool to allows us to actually uncover potentially bioindicator species and features that will impact on our environmental state of interest. And that I have just one slide of conclusion. We've shown that multimodal machine learning using eDNA and biogeographic features significantly outperforms machine learning using eDNA only, at least for impact prediction in this study. Differential abundant analysis achieved very significant data reduction, 90 to 99%, while preserving important features for machine learning. The best model was MLP with 83% accuracy, followed by tree-based model, and finally support by the machine. I need to mention that the results seems reasonable given that Francois' study using a smaller subset of the data, 69 sites, return accuracy of 69. Again, that's based on training data sets. So I would expect at the moment, realistically, as I shown in the first baseline, we were getting at around 60+% accuracy. That probably will be realistic outcome. And so it's very encouraging to see that with the various stepwise increment, we can boost the accuracy with the multimodal data set all the way up to 83%. I also note that some models, specifically SVM, adversely impacted by feature skills. So tree-based model have a clear advantage in the future because they are known to be less sensitive to feature variants. And Sharp analysis is powerful in that it can review key taxa and features impacting model predictions. And finally, the workflow I've introduced allows not only biogeographic data, but ingestion of other modalities in a tabular format. And that's all I have for the talk. It took a farewell, but thank you for listening.
Cheers. Thank you so much. I think it's so cool how you can combine environmental data and our eDNA, ASV data, and yeah, totally be able to model that. We are getting some questions through. So please, audience, continue to pop in your questions. Um, Katie Hillier has asked, fantastic talk, Josh. What advice would you give to non-machine learning experts to the analysis of eDNA datasets?
Um, I guess it depends, like because this is a context of machine learning. From the machine learning perspective, start with the automated machine learning packages, because that abstracts away the heavy lifting, the technical requirement to the package itself. And as I've shown, you could get quite reasonable, good out-of-box performance in a machine learning context. And from the bioinformatics perspective, again, as I mentioned previously, our group is developing a software, a GUI, user-friendly GUI, to to power through the entire, from raw sequences from the sequencing machine up to the point of you get ASVs, etc. So I think the eDNA research can be helped really by more user-friendly packages. So that's where I think we're heading towards. Yeah.
Yeah, it's really great to hear that there will be soon be GUIs that those of us who are less familiar with can use. Let's see if there's more questions. I just had a question. What do you think about like the 85% accuracy? Is that enough for managers? Like what would constitute, good enough in order to start making, say, management decisions with, you know,
I think that's case by case basis. So
mm,
not able to comment, but I think that's really the more important thing. I think is the reproducibility and
mm,
explainability, because regulators want to know exactly how do you come to this point, and can we reproduce this day in day out, season or cross seasonal, and if you can uh, first nail that down, I think it presents a method for consideration in any management decision. So I think ultimately that's case by case. Whether 85 is sufficient. Yeah.
great. Well, if there's no more questions, I'm sure you can reach Josh by his email. And yeah, don't forget to yeah, resubscribe to the Southern eDNA Society so we can keep bringing you quality content. Oh, yes, we get another question. Catherine has asked, you mentioned near the start of your talk the software Chimera or Chimera.
Yep.
Is that related to your machine learning work? And does it have to
Uh,
do it?
so it is not at this stage, but we do have plans to bolt in auto ML packages onto the. It just seemed natural that we present a package that could do the basic core bioinformatics function and extend it using AI or even machine learning packages. So really, we're just throwing everything potentially in the future to make a tool. That's why it's called Chimera. It's parts and bits and bolts of many things. Yeah. So
Great. I can't wait till that comes out. That sounds really amazing. Yeah. And do you know when that might be available to the general public to
I'm
use, or can we contact you for beta testing or?
Yeah, absolutely, absolutely. Really like to look. There is so much more. So essentially, this is just a tip of the iceberg in terms of machine learning. There is still foundation models and transformer models, and large language models that has yet to come, but will come. And the only thing is that I'm really interested in collaborating or working with anyone who's interested, have eDNA data sets and want to explore, some of the techniques. The package Chimera will be out hopefully next year. Yeah. And yeah, we're just in the process of fine tuning and making it relevant and etc. and those things. Yeah.
Awesome. Yeah, really looking forward to it. That'll be awesome. And of course, I'm sure you guys can contact Josh and get more details. I think that's all the time we have now. If you have any other ideas for who you want to see as webinar talks, feel free to email the Southern eDNA Society, throw your name in the hat, or if you've got a friend who does eDNA research and you want to see them talk, feel free to suggest them as well. Yeah, and thank you all for coming. Have a great day.
Thank you.
Cheers.